Okay students, today we are going to continue the genetics chapter of ICSE class 10 biology. This is the last part of this chapter. If you haven't watched previous parts, then you can get the video link in the description below and you can watch it. Okay, so let's start. So in this part we will study about the Mendel's laws of inheritance. Okay, so Mendel gave us three laws, according to which the first law is the law of dominance. Now what this law of dominance states? Law of dominance states that when a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. Okay, now how can you understand this? Let us take an example. Plant which is having the alleles capital T and capital T. As we saw that in the Mendel's experiment, we always start with the pure form of the characters. Okay, so it is having capital T and small t small t. Now if we will cross it, then in the F1 generation we will get capital T and small t, which is the dominant form of that character. Okay, so whenever we are crossing the pure forms of a character, okay, we will always get the dominant form of the character in F1 generation. So this is the law of dominance. Okay, so here we can see that capital T is present, which is suppressing the small t, as a result of which the character is dominant. Okay, next is law of segregation. So paired factors responsible for a character segregate into gametes and are recombined at fertilization. If we will see, when we cross capital T with small t, we will get capital T small t in F1 generation. Now if we will do the selfing, then what will happen? During the gamete formation, each gamete will contain only one allele, as means from this particular character, which is capital T and small t, the gametes will contain capital T, small t, and from here also it will contain capital T and small t. So these are separated from a particular gene, we can say. So this separation of alleles will recombine during the fertilization. Okay, this is called law of segregation. So what it states? It states that paired factors, we can say the factors like capital T is a factor and small t is a factor, these are responsible for a character. They will separate into gametes. Capital T will separate, small t will separate, and they are recombined at fertilization. Okay, so during the zygote formation they will again recombine. So this is the law of segregation. Next is law of independent assortment. So what it states? It states that the distribution of members of one pair into the gametes is independent of the distribution of other pair. How we can understand this? Let us take an example of a plant containing alleles capital T, which is for the height of the plant, that is tall, and capital Y, which is for the seed color, that is yellow. Okay, these are dominant, and here small t and small y, which is responsible for green color of the seed, and the height is short. So during the F1 generation we will get capital T, capital Y and small y, which is again the dominant form of the character, both height and the seed color. Now if we will do the selfing to get the F2 generation, here during the gamete formation, capital T can go with capital Y. Okay, so it will be capital T, capital Y, and capital T can also go with small y, so it can be capital T, small y. Okay, means which we saw in the dihybrid cross. So it is independent. It is not that it can only go with capital Y. It can go with any of the allele of the second character, or we can say the second pair. Okay, like capital T, capital Y and capital T, small y. Similarly, small t can also go with capital Y and as well as small y. So they are independent. They can go with any one of the other pair. This is the law of independent assortment. Okay, so these were the laws of Mendel's. Now the last thing which we need to know about this chapter, that is mutation. So mutation is the sudden change in the genetic makeup or in the structure of chromosomes. Okay, means whenever there is a change, like we saw in, we have already studied the structure of DNA in the cell division chapter. So there we saw that the DNA consists of genes. So if there is a change in that particular gene, okay, or the gene sequence, then we refer it to as mutation. Okay, so this causes many effects in the human body. Like an example is sickle cell anemia. Okay, sickle cell anemia, during this condition what happens, then like we see here, we can see that normal red blood cell is like this, but if a person is suffering from sickle cell anemia, then the shape of the red blood cell will turn like this. Okay, as a result of which it will not be able to carry sufficient oxygen. Okay, so this is an example. Another is radioactive radiations. So radioactive radiations are radiations which are caused by atomic explosions, like the explosion in Hiroshima, Nagasaki. So due to that explosion, what happened, there were radiations. So that radiation can cause deformities in the human body, which goes from one generation to another. Okay, so these are the two examples of mutation. So from this we have already completed the chapter genetics. In the next video we will start another chapter. So thank you for watching. We will meet in the next video.